So, first of all, I'd like to start off by extending our sincere apologies for having to move this presentation from Tuesday to today, especially to those of you who didn't check the calendar. But thank you for coming back. I'd like to welcome you. Pardon? Oh, sorry. I'd like to welcome everyone to the Physical Biosciences Division Science and Technology Presentation Series, and all those who may be viewing remotely. Today's topic is Microbial Sciences in the DOE Systems Biology Knowledge Base. Our speaker is Paramvir Dehal. Some of you may know he is part of the Virtual Institute of Microbial Stress and Survival and the K-Base Microbial Science Team Lead. Dr. Dehal started working in genomics and computational biology in 1998 when he led the comparative analysis of human chromosome 19 and the related regions of the mouse chromosome at the JGI. He worked on developing large genome annotation systems for the sea squirt, pufferfish, and general eukaryotic genomes. He's done extensive work in evolutionary genomics and phylogenomics, and since joining Berkeley Lab, he has focused on systems biology. There will be time after the presentation for questions, and please join me in welcoming Dr. Paramvir Dehal.
The intro, and thanks to all of you for showing up. My talk today is about the DOE Systems Biology Knowledge Base, and specifically the microbial science domain. I'm the lead of the microbial science section of the knowledge base, and so a lot of my discussion will actually focus on that. So I thought, like, just the outline of the talk itself, I'll give you a general overview of the K-Base project. I think a lot of you have probably heard of it, but aren't quite sure what it is or how it applies to you or your work. Then I'll talk a bit about some of the science that we've been working on, and then specifically the microbial science domain. And then I'll give an overview of some of our infrastructure, and then at that point I'll kind of diverge a bit and go into some of the things that we have under development, and close with some of our future work. So, the Department of Energy has been funding a lot of these very large efforts in genome annotation, sequencing, metabolic modeling, feedstocks for bioenergy, bioenergy research, a lot of foundational research. And a lot of these efforts have been focused very narrowly within their specific area. And all of this data, all of this information that's been generated by the sort of genome science program in DOE hasn't really been integrated that well. It's been kind of developed by groups in isolation, and the K-Base is an effort by DOE. It's an opportunity for all of us to try and integrate all of those different activities together. So DOE has a very, very broad mission statement, very kind of broad goals of trying to predict, control, and design biological components of energetic processes and environmental balance. And this is something that's just incredibly complicated, and just by the nature of it is going to require lots of different disciplines to come together to try and build these models and this level of understanding. And so the complexity of this mission, with the rapidly expanding and intricately related data types, really mean that scientists have to be able to basically augment their cognitive abilities. And so that's where K-Base comes in. And we're trying to help filter information, focus people's attention, ask the right questions, leverage other minds, and hopefully from the DOE perspective, make the impossible possible. So why is the DOE interested in this? Well, a lot of the work that we're doing at the lab across DOE is really focused on complex problems that are integrating across several domains. And in this team science focus, you need to have something that integrates all of their efforts together. So basically enabling the whole to be greater than the sum of the parts. So we want to increase the communication and decrease duplication of effort. And this really requires building communities. Accelerating the transfer of biological data or biological knowledge and analytical know-how. So being able to get information from one group, something that's been done under technology development in one science-focused area and transferring it to another. Stewardship of the knowledge, data, and tools. So when someone develops something and they move on to another project, that know-how, the tools that they've developed don't just go away, but they're maintained. And then solving the biological grand challenges for energy and the environment. So DOE really feels like they need to have an integrated process to be able to solve these grand challenges. So the broad DOE mission is really very, very complicated. We're talking about environmental cycles, carbon cycling, biogeochemical cycles, and how that impacts everything from sorry, I forgot to get a pointer. How that impacts cellular metabolism, individual microbes, communities, and basically ecosystems. So DOE is, of course, interested in this because currently a lot of the bioinformatics in DOE is very stovepiped into specific domains. It's not a very open system. It's hard to create working groups across different organizations, the national labs, the research, other research projects in the wider community, and really accelerating reproducible science where something that's been done can be done over and over again without it having to try and recreate something in someone else's lab. The reason why we're interested in this is we believe it's necessary to solve these grand challenges. We believe it's necessary to deal with these deep issues in scientific communication and reproducible science. Thanks, Ben. And we want to lower the barrier for computationally efficient use of algorithms and data from diverse producers. This basically just means that for people who are generating data, it should be relatively straightforward for them to actually analyze the data without having to become an expert on the computational side as well. And we believe in order to do this, we need to leverage many minds. So K-Base is effectively, a lot of K-Base is about community building. Defining what our data models are and pushing those data models through predictions and experimental design. Reproducible, reusable, transparent science. Enabling scientists to work together to approach complex problems. And then importantly, being able to give credit where credit is due. So when someone develops something or generates data sets, to actually be able to have that as a citable object and to be able to point to where that data came from or where that method came from. And let the person who created it have credit for what they've contributed. KBase is an open software, open data environment where other people can contribute, and we are completely open at this point, and other people can contribute to our code base. So what we really are trying to do is empower our users and allow the users to access our tools through multiple methods, command lines, programmatic APIs, and web browsers. Upload their data, use the analysis tools that KBase has developed, but very importantly, to kind of close the loop where have the tools and services that KBase provides to return to the users predictive models and hypotheses that they can then go back and test and then continue this process to improve our models and improve our predictions. So the initial platform of our module is separated out. We've got the GUI components up at the top and then the hardware stack down at the bottom. And we do this by having our hardware infrastructure, programming languages on top of that allow you to access our compute resources, data stores for large sequences or large data sets, relational databases, should be MySQL, and then infrastructure services to allow users to get at the core services down below, like the hardware. And then on top of all of those, we've built the actual computational biology services that allow people to annotate genomes, build metabolic models, assemble genome sequences, and then have those interact with our GUIs to allow people to get to access these services without having to learn too much about the details of the KBase architecture. So I mentioned that we have data stores to store diverse biological data, ranging from highly structured data to large bulk data sets that are potentially several terabytes large or in size. The central data model, we have structured storage for curated data. We've got a user data model to allow users to store their own data privately and share it with whoever they'd like. And we've gotten a sort of auxiliary data model that would store things like raw sequence reads that could be potentially several terabytes of data from a metagenomic sequencing run. The KBase central data model is not just simply a relational database. We've built data models for all of our data types and then encapsulated or captured that within our central data model, so that you can say something like a compound is a reactant of a reaction. That reaction is included in a metabolic model. A genome is modeled by a metabolic model, and something like growth phenotype could be predicted by that model. So all of these concepts are in there, not as simple or classical or relational database links, but actually as effectively assertions of how relationships work. So KBase has now been around for about a little over a year and a half. And on the science side of what we've actually delivered, in the sense of what can we do now that was hard before, we've created a system that transparently accesses multiple heterogeneous data sets. You can efficiently annotate a new microbial genome and infer the metabolic and regulatory networks. You can transform network inferences into metabolic models and map these reactions to genes using reconciliation tools. Designing efficient sequencing strategies for metagenomic projects. Test microbial ecological hypotheses through taxonomic and functional analysis. Predict plant gene function and phenotype via navigation and analysis of tissue-specific co-expression networks, plant variation and population genetics, and then importantly, being able to share data, analysis tools, workflows, and conclusions with the community. So I'll go through a couple of these examples for the microbial communities and plants, and then focus more on the microbes. So the communities group has created a set of tools that allow for automatic processing of metagenomic data, novel sequencing quality control, and evidence-based design of metagenomic experiments, so that you can come to KBase with a idea of what you're trying to do, and then KBase will help you design your experiment so that you have the statistical power to actually make the conclusions that you're trying to answer. On the plant side, an initial set of 7 genomes were targeted as important KBase or important DOE genomes, and the focus has been on trying to map phenotypes to genotype. So being able to take large-scale SNPs and phenotype data and then try and map those variations to specific regions in the genome. And then using the sort of variation workbench, you can select SNPs, see what the log odds are for associations with certain traits, and then follow up on those using kind of more classical genetics approaches. So on the microbe side, our overall goal has been to reconstruct and predict metabolic and gene regulatory networks to manipulate microbial function. To vastly increase the capability of the scientific community to communicate and utilize existing data sets and enable the planning of effective experiments in maximizing microbial systems functions. So what we've created is a way to link compute KBase services and data sources. So we've got user workspaces, relational data in the central store, and large-scale compute. So we can do assembly on large-scale computations, annotations, metabolic modeling, phenotype simulations, and phenotype reconciliation. And then this will give the user predictions of validation experiments that can then go back and help our annotation efforts. In the same model, we've been able to look at biological trends across the tree of life. So for all of the genomes that we have within KBase, we can automatically go through a process of querying the genomes and constructing models of those genomes, and then comparing the models to each other, and then having a unified core model of metabolism across microbes. So here I've sort of plotted some metabolic distance across phylogenetic distance to see kind of core metabolic units. And then additionally, we've provided services, or we've assembled our services so that users can troubleshoot problems in their wet lab, where if you have an example of trying to culture a microbe that you don't have all of the information for culturing it, we can take the genome, deconstruct it by looking at the annotations, do some metabolic modeling, incorporate the regulatory network, and then make predictions of what culture conditions would be valid for that organism, and then have experiments that could then go back and improve this model, and then hopefully by the end, we could be able to get a prediction of the media conditions necessary to grow these microbes. So a lot of this is focused on integration, and so one of our big projects was being able to take a genome sequence and feed it through this integrated pipeline where we have an annotation. That annotation generates a metabolic model. We reconcile that model with our experimental data sets, growth phenotypes, growth curves. We make regulon predictions, a regulatory model, integrate our metabolic model with our regulatory model. And then once we have our metabolic model, then we can start asking questions of our model, like making phenotype predictions, flux predictions, hypotheses like growth condition hypotheses, and then importantly, design pathways. So all of these are accessible to users through our web API, programmatic command line scripts through Iris, as well as a rich web GUI. So one of the things that you can use is a command line tool on the web, and through this series of about 9 commands, you can go through this entire pipeline of annotating a genome, building a flux balance model for it, setting some media conditions, running a flux balance analysis, and then comparing that flux balance model to known phenotypes, and then reconciling those phenotypes to the known data sets so that you can have a gap-filled, improved model. And you can do this through a command line. There's also a web GUI for this, where you can just kind of go through, click through this process, select the genome, select a workspace, build a draft model, set all of the media conditions for the model, and then basically generate a gap-filled model out of our tools. So once you've built your models, these K-based models, we have a workspace where you can kind of look at these and then share them with your collaborators. All of these objects can be versioned. All of them have full provenance of like what happened to generate that data set. And these metabolic models can be viewed through several tools that K-Base is providing, everything from simple tabular views to interactive metabolic maps. So outside users have also been able to use K-Base to integrate their tools. So this is an example of third-party tool integration by Nathan Price from the Institute of Systems Biology. He built a program, the Probabilistic Regulation of Metabolism, or PROM, that integrates metabolic models with gene expression networks to build a better prediction of growth phenotypes given the metabolism and the transcriptional network. So previously, it had been very difficult for people to use this. It was basically a MATLAB program that only his grad students had been able to run. And previously, it had been only applied to 2 organisms. And now that it's integrated with K-Base, we can run it on all of the genomes where we have expression data for. And we've been testing this on Shuanella MR1 with our transcription knockout and growth fitness data that's available in K-Base, basically validating his method. So across K-Base, we also have projects that go across our science domains, and networks is an example of that. So we have a network ontology and things that will facilitate building predictive models using our networks. And then this infrastructure for the same infrastructure also supports social networks for scientists. And we have network quality control tools. So some of the data sets that we have for microbes are actually quite extensive. So we've loaded several data sets from several sources, and we've got over 5,000 sources or data sets, and we can use our API to build heterogeneous networks in a unified format for gene-gene networks, gene clusters, mixed networks, or integrated networks. And here's protein-protein interactions and regulons. So if you go to our KBase homepage, which is kbase.us, I should have written that down on the slide somewhere, but if you go to kbase.us, you'll see our homepage, and we have a link right in the middle that lets you download the tools that we've built or to try KBase right now. We also have a third way for you to interact with KBase, and that's through our labs projects. So KBase labs are prototypes and applications that demonstrate KBase present and possible future functionality. So early stages of functionality, we kind of provide a space for users to come in and provide feedback. So we've got, I think right now we've got something like 10 or 12 tools that people can kind of click on and play around with. We also have extensive documentation and tutorials. So if you go to our website, you can follow a tutorial to do any number of things, from annotating genomes, building models, analyzing metagenomes. So that's sort of how KBase has been organized right now in this first 20 months or so of our project. But now we're starting to get more into how we want KBase to sort of work in the future. And one of these things that we're trying to design right now that we don't actually have released yet is the concept of a scientific narrative. One of the things I said early on in the presentation is that one of the goals of KBase is to facilitate sharing and facilitate that sort of cognitive process that someone goes through when they're analyzing their data. And the way we're looking at it is through this process of defining a narrative, where you kind of go in with your data set and you describe what you're doing as you're doing it. And then someone else can look at your narrative of how you've done something, comment on it, and provide feedback nearly in real time. And so users can then share knowledge at all levels of granularity. We have the capability of having project management, where the creation of teams and sharing across teams, you'll know what's going on across the project that you're working on. These narratives, when they're finalized, can be reviewed, assigned DOI numbers, and potentially accepted as a journal publication itself. We can incorporate metrics of research efficiency, how long the time it takes to complete a narrative, times from hypothesis to confirming data narratives, as well as building a lot of narrative navigation tools and sort of user and algorithm rating for the narratives that people create. And this sort of goes a lot towards the social component that we have for KBase, of allowing users to follow other people's research, create their own narratives, look at the narratives that they have in development, and then whatever data and data stores they have and the people and projects that they're participating in. So in this concept, we have an interactive community that's being generated, or that's being facilitated, where these narratives provide a measure of activity and influence. The functions and data content can be tracked, and all of our narratives, we have code versioning, scenario branching, so you can share your narrative with someone else. They can take your narrative, apply it to their data or their system, make changes that they need, and then share their changes to your narrative back to the community. So I think I'm going to get a little bit more into, or I just want to shift a little bit more into talking about some of the K-base philosophy. And K-base has services and data, but K-base isn't a workflow engine. It's a sort of part of K-base, but it's not really what K-base is about. And K-base is not about creating analysis pipelines. I think, like in my microbes talk, I talked a lot about allowing users to go from a genome and phenotype data to a reconciled model, and that is a lot like a pipeline, but that's not really what K-base is about. What we want to do is enable predictive biology. That's the goal. We want people to be able to create models that demonstrate understanding of biology. We need to have these models backed up by data and software that has complete versioning and provenance, so we know what went into our model, so we know where our models work and where they don't, whether or not they were based on some flawed assumptions, whether or not something gets invalidated because the data the data was just somehow flawed, or the software methods were somehow flawed. And we want to capture the user's creative process. So all of those things that you do when you're looking at your data set and you're trying to analyze it, there's a process that you go through when you're analyzing it where you think to yourself, well, I wonder if this is what it looks like. Maybe, maybe not. You try something, that turns out to be wrong, so you try something else. And you go through this large exploratory process where sometimes you just have a hunch and you think, this must be right. Let me go down this path for a while and then see where I go. And this is very different than what it looks like when you read a paper. When you're reading a paper, you're only reporting the path that worked. You're not talking about the dead ends. You're not talking about all of the other things you tried. So we want to capture that creative process. And so one of the key concepts here is structured assertions that we want to help users make. And these structured assertions are computable biological assertions on K-base entities. And that's sort of like a strange way of saying that we want people to make assertions that are machine readable so that a computer can read someone's assertion and know what it means, what sort of biological claims that assertion is making. And then another important quality of the assertions have to be that they're falsifiable. You can't, it's sort of meaningless to have these sort of vacuously true assertions or assertions that just can't be falsified, where you say something about, I think an unidentifiable compound is triggering cell growth. If it's unidentifiable, it would be hard to prove that you can't identify it. We need these assertions to be linked to the supporting evidence and to have confidence values when they exist. So sometimes you have an assertion that has supporting evidence, and other times those assertions don't really have that much evidence behind them. But we still want to capture that assertion. And when you have a confidence value, you need to be able to assign it to that assertion. To that assertion. And because we want these assertions to be computable or machine readable, we want to be able to do automatic identification of incompatible assertions, a sort of consistency check across all of the assertions that exist in our knowledge base so far. So this would tell you like what assertions are compatible with the assertion that you're making, which assertions are incompatible with your assertion. And obviously because it's going to take a lot of people to write these assertions, we want these assertions to be capable of being mapped across versions, mapped to related features, and even mapped to other assertions themselves. So these assertions should be structured in a way that we can actually transfer them. So with all of that, these assertions still have to be precise enough to be meaningful, but they have to be lightweight enough for people to actually take the time to make them. So it can't be so burdensome that people won't bother doing it. And then, controversially, we want these structured assertions to have user ratings that go along with them, where other people can comment on other people's assertions. Comment in the way that I think this is right or I think that's wrong, but hopefully something more precise than that. All right, so let me give you an example of what we want to capture. And this is some text that I got from another researcher in PBD, Morgan Price. And I'm just going to read this really quickly because it's sort of important to how this process will work. So Morgan writes, the G20 genome contains 2 paralogs of HDR, heterodisulfide reductase, Flox operon, and Flox is flavin oxidoreductase, HDR Flox 1 and HDR Flox 2. HDR Flox has never been characterized, but is believed to be a cytoplasmic electron transfer complex comprising HDR CVA and Flox DCVA. DVH has a single HDR Flox operon DVU gene list, with DVU 2401 being a Flox DC fusion. Flox ABCD might be involved in electron bifurcation between ferredoxin, NADPH, and the HDR complex. And HDR ABC might send electrons to DSRC. Although HDR Flox 2 has few phenotypes, HDR Flox 1 is very important for fitness on formate sulfate and is detrimental to fitness during co-culture with a methanogen. So that's a pretty dense statement. And it's hard to just read that and abstract out all of the things that Morgan meant when he wrote it. But it's obvious that there was a lot of work that went into actually generating this. This single paragraph here, I haven't checked with Morgan, but I would guess that it represents several tens of hours of Morgan's time staring at the computer, looking at data sets, reading papers, going to Metapsych, seeing if these things match up right, and then looking at data to see if it actually fits the way he thinks about it. And the data sets that he's using to come to these conclusions, those represent several hundreds of hours of researcher time in generating those data sets. So several RAs in Adam Deutschbauer's group generated that data that then Morgan looked at in order to produce this one paragraph. And so obviously it's very important, so it needs to be shared. So of course you can find it. It's found at morganprice.org/temp/G20/reannotation.txt, which is a pretty obvious place for it to be, obviously. obviously, you would know immediately to go there. But in this text, how many assertions are actually in this statement? What are the dependencies of these statements? Like, how does this sentence relate to the last sentence? Is it truly dependent on each other, or are they independent statements? Are the assertions falsifiable? What's the confidence that we have in these assertions? What does might mean? So what we want to do in KBase is to actually take this sort of thing and capture it in a more systematic way. We haven't actually done this yet. This is one of our projects that we're going to start in the next couple months, and hopefully have a couple prototypes out somewhere at the end of the summer. But if you deconstruct the statement a bit, you can say, like, all right, this statement basically becomes G20 contains two paralogs of HDR flux operons. HDR flux is a cytoplasmic electron transfer complex, and you can link it to your confidence and link it to your evidence. DVH has a single flux operon. DVU2401 is a flux DC fusion. Flux ABCD is an electron bifurcation between ferredoxin and ADPH and HDR. HDR sends electrons to DSRC, and here you would encode the might into your confidence. HDR flux 2 has few phenotypes, so this is kind of a strange assertion to make, where it's only has a few phenotypes for the data sets that we have access to. It's hard to say that this is just globally true no matter what. So this probably needs more qualifiers, and the evidence here is probably just some small set of experiments that have been run. But nevertheless, we still would want to capture an assertion like this. And then so on. You can kind of go through these more. But, and then deconstruct all of these to things like HDR flux 1 is detrimental to fitness in co-culture with the methanogen on lactate. So all of that text that we just went through is just that little part of this entire document that Morgan has on his website. And it all reads just like that. So there's this insane text document with no figures or anything, just 4,600 words of text, incredibly dense. But it's not something that's published. It's not something that would necessarily go into a paper. You wouldn't necessarily publish all of that. You might find one or two stories in there and have like a really nice paper on some aspect of this organism's energetics. But the rest of that information is not going to go into your publication. It's just going to sit in Morgan's notebook or in /temp on Morgan's website. And if someone wants to get access to it, they have to know who to ask. So with all of these assertions that we were able to pull out of Morgan's text, we have to be able to now compute on them and to be able to ask, if I have a new data set, is my data set consistent with the assertions that have already been made? What do these assertions say about related organisms? This is all about Desulfovibrio G20. What about other closely related Desulfovibrios, or even something that's more distantly related? Can I compose new assertions based on the assertions that Morgan has made here already? Like I could look at this statement up here where he says G20 contains two paralogous HDR flux operons. And then he has the statement down here, flux 2 has few phenotypes. Well, maybe I would predict, just based on this, that flux 2 is duplicated, but it's non-functional. It's some sort of pseudogene, or a pseudogene cluster. And then I would want to be able to assign my evidence to it, or maybe I would want to investigate it further before I just push something like that out as an assertion. And then, like I said, I want to be able to compute on it. I want to know if I can take a statement like this, where flux ABCD is involved in electron bifurcation, and HDRC sends electrons to DSRC, and I want to know, like, given these two assertions, is my statement down here correct? You'll notice that Morgan's statement up here isn't necessarily that precise, but I've actually written out an equation down here, and actually I'm just now noticing he has NADPH and I have NADH, but maybe that's still okay, I don't know. But is this statement consistent with these two statements? I want to be able to do that automatically. I want KBase to be able to tell me that my assertion is valid or not based on the existing assertions. And if it's not valid, then maybe I just don't believe Morgan's assertion, or maybe my assertion's wrong. One of them has to be right. So generally what I want to be able to do is to make these, to use my structured assertions to make predictions. I want to be able to go from conditions to activities. And so one way to do that is to just have a very simple conceptual model. So you have part of it is what is it? What's there as part of my model? And in this case, HDR flux 1 is what's there. Where is it? It's in the cytoplasm, and it's some sort of electron transport complex. How much is there? Well, in this case, I don't know how much is there. Some is there. Who does it interact with? Well, we predicted that it interacts with NADPH, it interacts with ferredoxin, and potentially DSRC. So now that I've defined what it does, I should be able to state some conditions and then predict some activity from it. So if I say, growth on formate sulfate, this prediction should say the activity should be something like growth on formate sulfate. If the conditions are co-culture with a methanogen, with lactate as the carbon source, maybe the activity is very low. There's little growth. And so if I have models like this, I should be able to then expand my assertions out to other fields that don't necessarily need to know the details of the actual energetic balance, but just want to know at a higher level of like the microbial ecology of if I have a methanogen and I have G20, what are the relationships there? You can get growth based on those conditions, and if you need to, you can step down into that model of why it's important, of how the electrons are actually moving. And this simple conceptual model would be components of a larger process that you would use to build up all of these models and then finally have something that's more like a genome scale model. Okay, so so this work is stuff that we're working on right now, and we're trying to generate that language of how structured assertions work, trying to figure out how we want these assertions to relate to each other, how to move from these assertions to simple models, and how to move from those simple models to larger genome scale models. But in the future, we also have several things that we need to work on. We've got to make sure our data model evolves to support our modeling mission, a framework for tuning algorithms output into models that need further development. We have to have a theory for integrative cross-scale predictive biology. So something that actually unifies all of what we're doing. Better ontologies for more or less all of our data types. We need to improve the data import and quality assessments of metadata. efficiently incorporate new third-party algorithms, grow an external development community so that the K-based developers aren't the only ones who are contributing code, and come up with a licensing scheme so that we can truly be open source. And then also build the social tools. So this would be a more concrete view of that narrative interface that we want to build. And with that, I'd like to acknowledge the K-Base team. This is a very large group across several institutions. There's about 50 people who work on K-Base. So it's a pretty large effort from several national labs and universities. So with that, I'm happy to take any questions. Yeah. So your question was whether or not we have international collaborators?
Yeah, if you have international collaborators.
Sort of. We have nothing formal. We just have international people who contribute to K-Base, but none of them are funded through K-Base. So people like, I know that Dylan Shivian is working with Gene Tyson, and I know that Volker Meyer has a lot of collaborators in Europe as well, but nothing formal. Well, like across these institutions, there's actually a formal agreement of being part of K-Base, but in the nature of K-Base as being an open project, anyone can contribute to it. Oh, sorry.
I have a couple of questions. I went to your website and I tried using your tutorial, and I got to the point of looking for 16S RNA, which I'm interested in, from cyanobacteria, and I was told that the connection wasn't ready. Is there any prediction as to when this is going to be operable?
If you were following the tutorial, it should have worked. I know that we're in a transition right now where we just moved a lot more code to production, but the tutorials, if you're following the tutorials, all of that functionality should still work. I will definitely look into it if there's.
Okay. The other part is that I've been using for a couple of years now databases at JGI and at the NCBI. And I find that the data that are coming in at an enormous rate for the cyanobacteria, for example, are literally shot full of holes.
Yeah.
And is there any way that that's going to get remedied?
So that's one of the things that we're trying to work on by pushing users towards these models, where if you take the cyanobacteria, for instance, there's a lot of there were a lot of holes in metabolism. There's a lot of
well, just in the genes, you know.
Yeah, in the genes, the
gaps.
genomes just aren't
and
complete.
they're described as complete genomes.
Yeah. So that, unfortunately, all we can do is point out where the holes are and why those why we can't make the full set of predictions, because the holes in the annotation, the holes in the physical genome assemblies, and effectively like kind of score it as low quality and then kind of tag it for biologists to go back and try and fill in those holes. And what we want to be able to say is, because the data is in this state, predictions like these cannot be made using that data. And if you want to make these predictions, you need to collect data that looks like this. So a lot of, for the cyanobacteria, we have a lot of growth phenotype data, like just growth in different media, different conditions. So we can infer a lot of what genes and functions ought to be there, but where we fail is actually mapping those functions to genes because there's holes in the annotations. But hopefully in this framework, it'll be easier for people to collect data.
Actually, my question was along the lines of the previous question, and what I wanted to understand is where your models are and how many case studies that you've been able to take the model to predict what would happen and then verify that your model actually predicted the what actually happened.
Yeah, that's a good question. So we have the capability of building things like metabolic models and regulatory models automatically for all of the genomes. So those basically exist as soon as as soon as a genome is deposited into KBase. But as you might guess, the quality of that model is not that great. So what we've been focusing on is building tools that take data to improve the models. So things like phenotype data, mutant fitness data. If you had additional data that would say something about the regulatory models. And in one case with Shwanella that we used as a sort of target organism that we worked with Adam Deutschbauer on, we were able to automatically build the model using his growth data phenotypes. And he had over 200 media conditions and something like 300,000 individual mutant fitness data types or data sets. Or 300,000 individual mutant data points. And then we incorporated those results into our model. And then when we used the model and ran it on different media conditions, we could predict, we predicted functionality that we thought was there and tried to assign that functionality to genes that was missed. And in a couple cases, Adam Deutschbauer was able or he had already verified that functionality was associated with the gene. But we were also able to make predictions about what other conditions they could grow on and just generally improve the model. And that's an example. That's also an example where the quality of the model is highly dependent on the quality of the data underneath. And so having examples like that and being able to say, predictions of this quality require data of at least this quality.
So do you actually use data from the literature, going along the lines of, how good the model is depends on the data that you use to calibrate the model. So do you use the internal data or do you use all kinds it sounds like these are very complex, so you are going to need a lot of data.
Yeah. So this is actually a very complicated thing you're asking. KBase incorporates some data that's just available publicly, but it doesn't incorporate all the data. A lot of things don't have a data model underneath. And for KBase to incorporate some type of data, we need to have a data model so that we can compute on it. So a lot of the data that we have, because there isn't like some repository that we're pulling from, that data is basically associated with a user. So if you have growth phenotype data, you may have some growth curves or you may have a biolog plate. You can store that data in KBase in your user space, and you can share it with whoever you like. You can make it public, and it's available through KBase. But we don't do anything to explicitly go out and pull that data in. So you can really do whatever you want, and you are effectively vouching for the quality of the data that you're using. So if you read the literature and someone just reported that my cyanobacteria will grow on light and this carbon source under these conditions and these nutrient conditions, you can just code that up by hand and put that in. We don't require you to give us the image of the the OD readout. So all of that data can live inside your own workspace. I don't know. Not sure if that answers your question.
So I'm primarily interested in sequences and annotations. Is my understanding of it that you get that information from another database, or do users upload that themselves? In other words, will you be any better than NCBI in that respect, or do you also pull that information from them?
So actually, we're hoping to be better than NCBI.
So you have your own set of curators, and
we have our own set of algorithms to do gene calling and gene function assignments. And importantly, these algorithms are able to learn from previous sets. So when I was talking about like these structured assertions, we would want those assertions to map forward to new annotations. So if you assign a gene function, that would inform the next iteration. For any type of user annotation, you can always define a genome and your own annotation set in your user space. So you don't have to use the K-base annotation or the NCBI annotation or JGI's annotation. You can pull it into your user space, alter the annotations in whatever way you want, either systematically through some code or by hand editing it, and then it'll live in your user space and can be shared to other users through there. But as far as the process goes, when we load a new genome, we do run our own analysis methods on it, and we keep it continually up to date by running those methods at regular intervals. So with NCBI, with RefSeq, for example, RefSeq's effectively frozen at submission time, and it's up to the submitter to ever change those annotations.
And maybe an unrelated question, but I was a big fan of the KEGG database before it was paywalled. Do you have pathway annotations as well? Okay, and is it like in a flat file that, like, suppose a more advanced user would want to be able to download and have their own SQL database and run queries on their own? Is it that accessible?
Yeah, all of that is accessible, and we even go a couple steps further where we have services that will allow you to just directly interrogate it, and we have APIs that, for several programming languages that you can just include or client APIs and then program against it.
Are there any other questions? Okay. Thank you. Thank you all for coming. We have another presentation on May 21st with Ming Hammond. Hope to see you there.